Okay, so now we're going to talk about carbon, and carbon, of course, is basically O chemistry, organic chemistry. So organic chemistry is the study of carbons, and we're just going to go over a few of the concepts that are important, of course, for biochemistry. And so organic compounds have carbon in them. They also have other elements as well: hydrogen, oxygen, nitrogen, sulfur, and phosphates. Now, carbon is going to be important because it can make four covalent bonds. It can also bond to itself, and so it's called a catenation. There are different forms of carbon groups. We have our linear groups, aliphatic groups, or we have our round groups, our rings, our aromatics. Now, carbon in life is very important because carbon molecules have functional groups, and these functional groups are going to react. And so these reactions by these functional groups are basically what makes life happen. So a few things about these groups binding to carbon. We have hydrogen, which bonds to carbon. So a carbon-hydrogen bond is inert, meaning not reactive in a biological sense. And then you have carbon can bond to oxygen, nitrogen, and sulfur. And this makes most of the functional groups. Now carbon is going to have hybridization states. And that means that it's going to form different number of bonds. So it can form 4 bonds. It can form, and these will be single. It can form 1 double and 2 single. It can form 2 double bonds. Or it can form 1 triple plus 1 single bond. So these are the different hybridizations that carbon can do. Backbones of carbon, just really quickly, carbon is very flexible in how it bonds, and so we have carbons that can form straight chains where you have carbon one after another. Carbons can branch, so we have branches that are going to be made by carbon, and then of course our ring structures, so they can form a cyclic ring structure. Now, how many bonds can carbon make to some important elements in life? Carbon can make, of course, 4 possible bonds to carbon itself. Carbon can make up to 3 bonds with our nitrogen, 2 bonds with our oxygen, 2 with sulfur, and of course, hydrogen can only make 1 bond. So the bonds that our carbon are going to form are going to be basically 2 types. We have our intra, which means within the same molecule, and then we have our inter, which means between species. And so for intramolecular forces, one of the more important ones are covalent bonds. For intermolecular forces, you're going to have also important hydrophobic, hydrophilic, and van der Waals. And some of the van der Waals forces can actually also be intramolecular forces. So van der Waals are going to be not true bonds. There is no sharing or exchange of electrons. Let's talk a little bit about our van der Waals forces, just really briefly. There are ionic bonds, hydrogen bonding, dipole-dipole, and London forces, or dispersion forces, and they're in the order of decreasing strength. So this is strength. This is London forces that are going to be the weakest, and this is the strongest. I'll change this one to weakest. Okay, so ions, let's talk about that. So these types of forces are ionic interactions are going to be both for and intramolecular. And they can form metal, they can involve metal cations. They can involve sodium, and they can involve chloride. And so they are going to be the strongest van der Waals forces. Um, Next to them are going to be the hydrogen bonding. So hydrogen bonding is a type of dipole. It's a very strong dipole. And the hydrogen must be bonded to an oxygen or nitrogen. And then, so this is going to be a stronger interaction than other dipole forces. And again, this can be both an inter or intramolecular bonding. Now, next up are our traditional dipole-dipole. And so these are going to be between polar regions of molecules. Okay. And so in this case, we're going to have partial charges. So you're going to have a partial positive charge, and one is going to have a partial negative charge. And those are usually given by a delta signal. And an example of this, of course, is hydrogen. We often write hydrogen with the hydrogens having partial positives and the oxygen having partial negatives. And then finally, our London dispersions are going to be very weak. They're short-lived. And it's basically the nucleus of one atom is going to be attracted to the electrons of another. So really in biochemistry, London dispersion forces are going to be important for nonpolar interactions. Okay, so this is going to move us into a concept of water loving and water hating. So hydrophobic is going to be water hating. Hydrophilic is going to be water loving. So when you talk about hydrophobic, it really depends on the amount of carbon-hydrogen inert bonds. So remember, CH bonds are not very reactive at all. So hydrocarbon regions are nonpolar. So that's when you have a fat with a long chain of carbon hydrogen. They're not water loving. They're going to be hydrophobic. So basically, your hydrocarbon regions are going to be attracted to nonpolars, and those are going to be by those London dispersion forces. So you can imagine that this is going to be important for things like your lipid bilayer, where you have a lot of the fat in your cell membranes interacting with each other. They're all going to be hydrophobic. Now, one thing to keep in mind is you're dealing here with carbon hydrogen, but we know that in life we have other elements that are going to be involved in molecules. And so basically, how do you treat a nonpolar carbon hydrogen bonds? How do you treat the water phobia, the hydrophobicity? And you do that by adding other atoms. So you add other atoms. And so oxygen and nitrogen are going to make a region very polar, and phosphate also. And then sulfur is going to still make it polar, but less polar than oxygen, nitrogen, and a phosphate. But still more polar than a traditional carbon-hydrogen bond. Now, hydrophilic, meaning water-loving, your hydrogen bonds and ionic interactions and dipole-dipole interactions are all going to play a role. So we have our H bonding, ionic, and dipole. And so molecules, some molecules will have both of them. So they're going to have hydrophobic plus hydrophilic. And these are going to really be important to the behavior of the molecule. All right, so just a little bit about bond strengths. So again, we know that ionic bonds are much stronger than hydrogen, than dipole, than London dispersion, of course. And we also know that all substances with an oxygen, nitrogen, phosphate, or a sulfur have a polar region. So the hydrogen-carbon region is going to be nonpolar, and it really, if you're going to compare two molecules, it's going to depend on how much of the nonpolar inert hydrogen-carbon bonds versus the other bonds. So let me give you a few examples. So if we have CH3COOH, and we have CH3CH3CH3CH3, then you're going to have, this will be more hydrophilic. And this, of course, will be less. And another example, of course, is going to be if you have the same number of carbons, but you have more oxygen, nitrogen, or phosphates, then you're going to have this one is going to be more hydrophilic. Oops. And this would be more hydrophobic. Okay, so now our hydrogen functional groups, again, hydrocarbons are not very reactive, but if you increase the number of bonds, they become more reactive. So this is less reactive as an alkane, and as you go across, this is more reactive. And this is going to an alkyne. Now, alkynes are going to be very rare in biological systems in our bodies, so this is rare. And the alkanes and the alkenes are going to be more common. Now let's just go through some hydrocarbon functional groups. So we have here, with respect to hydrocarbons, we have aliphatic, and we have aromatics. And alphatics, of course, can be in an open or closed. Aromatics are only closed. We also have saturated or unsaturated. And for the aromatics, we have unsaturated, and it's based on benzene. Which is a 6-carbon. The different names, you have alkane, alkene, alkyne. On the side of the aliphatic, the aromatics are going to be arenes or aromatics. And then some examples are benzene, toluene, and over here, of course, is methane, propane, butane, etc. So moving on to functional groups of oxygen and sulfur groups. All right, so here we have oxygen can make a phenol, which will be a cyclic, where you have an oxygen attached to the aromatic, and that's referred to as a phenol. We also have our double bonded and single bonded. So our alcohols will have a single bond with an oxygen. Our ethers will have a single bond with our oxygen. We're going to have our double bonds. So here we have our ketones, and then our aldehydes will have a double bond with oxygen. And then we have both single and double bonds, such as with our ester. So here we have our single and double, and with our carboxylic acid, also a single and double bond with our oxygen species. Now, one thing to point out is our carbonyl groups, which are going to be important. And so it's basically the carbon double bond to an oxygen. Carbonyl groups. And then, of course, sulfur on the other side. Sulfur is going to be able to replace oxygens in an alcohol or ether. So over here we have our alcohol and we have our ether. And so these two compounds over here, we actually make that a different color, not to confuse the carbonyl. Okay, and so you can replace that oxygen with a sulfur. And so those are going to give you a thiol group. So here's a sulfur that replaces an oxygen, and is called a thiol. And then we also have a sulfur that is going to be a thiol ether, squished in between two carbons. And then two of the thiols can come together to make a disulfide bond. Disulfide bonds are going to be important for linking peptides together, for example. And we'll see that with insulin. Nitrogen can form functional groups. You have your nitrogen can form an amine, an amide. An amine is going to be found in amino acids. Amides are in proteins. And here we have just showing two different ways. We have amines are derivatives of ammonia. And you can see ammonia has 3 hydrogens attached. And so basically you are going to replace those hydrogens with some sort of group. So for a primary amine, I'll leave it blue, you're going to have one group replace one of those hydrogens. For a secondary amine, you're going to have 2 groups replace 2 hydrogens. And a tertiary amine, you're going to have all 3 hydrogens replaced by some group. On the other side, we have an amide, and amide are important for our proteins because proteins are made up of amino acids. So here we have amino acid 1, we have amino acid 2, and you can take the carboxylic side of one amino acid to the amine side of a second amino acid and join them together to make what's called a peptide bond. And so this is 2 amino acids coming together, so it's a dipeptide. Another way to write peptides is in the form of mers. So there's 2 amino acids, and so it's called 2-mer. So if you have a 6-mer, that would be 6 amino acids. 10-mer, 10 amino acids, etc. Okay, now look, moving on to phosphorus. So phosphorus is going to come from part part of the phosphate comes from phosphoric acid. My computer just froze. Okay, so hopefully it's still going. So phosphates are going to be important in teeth and in bone. And also, of course, phosphate is going to be our energy. ATP, GTP, etc. Phosphates are going to be alone or part of a ditri. We have ATP, triphosphate, ADP, diphosphate, AMP, monophosphate. So here we have two different ways that phosphates are going to play a role. We have in which we have a phosphate surrounded by our oxygens. We have phosphoric acid. This can become a diphosphoric acid when you have two phosphates, or a triphosphoric acid. You also can form an ester with your phosphates. Here's a monophosphate ester in which you only have one R group, a phosphate diester in which you have two R groups, or a phosphate triester in which you have three of those groups attached. Now, alcohols are also going to react. So alcohols are going to be oxidized, which means they lose an electron or gain oxygen or lose hydrogen. Any of those apply. So primary alcohol, you have the OH group is attached to the terminal or end carbon. And so this can lead to an oxidation. And then you'll get an aldehyde. And this can be oxidized. And you can get carboxylic acid. Now, a secondary alcohol through oxidation will lead to a ketone. And an alcohol, a tertiary alcohol in which you have, again, we're counting these R groups, a primary, we have a secondary where you have more groups attached, and you have a tertiary. And in a tertiary alcohol, there's no oxidation. Okay, now with our aldehydes and our ketones. So our aldehydes can go through an oxidation. And give carboxylic acid, as we just saw. They can also be reduced to an alcohol. And we just saw that in the previous slide as well. The ketones can be reduced to an alcohol. And they really do not get oxidized, especially in a biologic system. There is no hydrogen attached to the carbonyl carbon, and therefore you cannot oxidize, remove electron, add oxygen. And unless you have a very strong oxidizing agent, which in biological systems we do not. Now, another thing is our carbonyl group. Remember our lovely carbonyl group? That was what I did in red. So here's this lovely carbonyl group. We had it on ketones as well as your aldehydes. And now our carbonyl groups plus alcohol, and it can go in both directions, are going to give you your hemi ketols or hemiacetyl groups. And this is basically, I'll write the acetyl version. And then you can add, again, alcohol. And it goes in both directions. And this is where you get your acetyl or ketol. And so the difference is you're going to have one group attached in a hemi and two attached in a full acetyl or a ketol. Now, reactions with carboxylic acids. Carboxylic acids can interact with bases, alcohols, or amines. And a base will give you a salt. And the salt, your N, is from your amine, and your carboxylate is from your acid. Your alcohol is going to give you an ester. And your amine is going to give you an amide. Now our thiols are going to give us our disulfide bonds, and so this is going to be through an oxidation. And it can break down into two thiols through reduction. And then amines are going to be important bases. They can react with acids. And so here you have an amine, you have your ammonia with a hydrogen and acid. Here's our acid, and it's going to give you your ammonium ion. And so medicines are going to add amines to make them more soluble. To increase solubility. Phosphoric acid is going to be similar to our carboxylic acid. So carboxylic acid, phosphoric acid are our two main important acids in biochemical reactions. And these are going to be able to form esters. And so you can see that you have can have 1 to 3 organic groups added, and they're going to replace the hydrogens. So you have 3 hydrogens here, and these can be, hydrogens are replaced with organic groups, carbon. And so you can have a monoester if 1 hydrogen is replaced, a diester if you have 2, or a triester. All right, so just really quickly, reduce versus oxidize. So if you ever forget which was which, always remember this. Carbon, hydrogen is the most inert molecule, carbon surrounded by 4 hydrogens, and that is the most reduced. And so again, if you forget, always go back to your CH4 molecule and think, okay, that's the most reduced. So let me look at my compound now and see how much other stuff is added to it. So when you add oxygens, of course, you're going to increase the solubility. You're going to make it more oxidized. Double bonds are always going to be more reactive than single bonds, etc. And so you have more than 1 oxygen here, so that's going to be more oxidized. The more carbon-hydrogen, of course, the more reduced. And then finally, polarity. Again, you have charges that get dispersed. So the polarity and the sharing of these charges determine whether or not you have a polar or nonpolar. And the more of these other elements you have other than carbons and hydrogens, the more polar a molecule becomes.